Hello, this presentation on the immune response is basically divided in two different topics. They are evidently related, that's why they're in this single presentation. So I first will explain briefly what triggers the immune response, how are the immune cell specialized, and how are they differentiating and maturing. And then the second part of the talk is basically how are the different immune differentiated cells characterized in what we call Th phenotypes or T helper cell phenotypes that have been identified with very specific types of immune responses and also have their associated diseases. So I'm calling this sustainability and specialization for survival because, after all, the immune system is there to help us survive and deal with danger, although sometimes it does cause disease.
The first step in the immune system response to danger signals is an antigen presentation. This antigen presenting cell, depicted here as a dendritic cell, travels in the bloodstream carrying pieces of the antigen that is perceived as danger signal, and they become in contact within the lymphoid organ with the naive T cell. This naive CD8 positive T cell will identify the antigen through the T cell receptor depicted in green, and the signal from the dendritic cell or the antigen presenting cell that tells the T cell this is a true presentation of a danger signal is the chemokines that are co-stimulatory chemokines, depicted here as the XCL1 and corresponding XCR1, the receptor to that ligand, on the T cell. This is what really tells the T cell that what they're identifying via the T cell receptor here, depicted in green, is real. They should respond to it. And the first response is basically by short-lived effector cells. These are mostly going to die very quickly, but they are the ones that are going to be like the first line of defense in this recruitment. A subset of cells are actually the ones that we know are called memory precursor effector cells. There's less of these, but they will be remaining in the lymphoid system in case the memory is required for future contact with the antigen. But so far, the dendritic cell has come from the infected site to the lymphoid organ and just sort of given the alarm to the T cell that now has been primed to respond. In a next step, there's a preparation of these T cells by expanding these clones that now have recognized the antigen. They are still in the immune system, but they're starting to tell each other they have to be ready to fight the antigen. And they also have to differentiate. The differentiation takes different steps. The interleukin-2 signaling will actually stimulate the short-lived effector cells for differentiating above the memory cells. In a first step, they're telling them to be ready to the attack very quickly. On the other hand, in the environment, depending on the type of antigen, the type of antigen-presenting cell, and also the type of co-stimulatory signal, we have now a differentiation that if driven by IL-12 or interleukin-12, it will produce a phenotype on the T cell which is called Th1 phenotype, characterized by the secretion of interferon gamma and TNF-alpha. On the other hand, if it's driven by IL-21 or interleukin-21, it will trigger basically an IL-17 or Th17 response. IL-27 I will mention later because this is a very important pathway that will trigger a control signal by basically secreting interleukin-10. But I'll talk about this at the end of this presentation. The important thing is not just the differentiation, but also the activation of these T cells. And the way that they become activated is they also will actually express in the surface the migration chemokine. These migration chemokines are going to be critical for them to now go to the site of infection, because so far everything has happened within the lymphoid system. There was a signal of danger that was presented by an antigen-presenting cell. This is like in a fire. The signal has come to the fire station, but the Dalmatians have to actually drive the fire trucks to the site of the fire to deal with it. So now we have the chemokines that are expressed that tells us those T cells have been now activated, they have been differentiated, and they expanded, and they're ready in circulation. They travel, and what they do with that chemokine expression is that they actually will identify the corresponding ligands of the very specific cells that are infected. And this all has to do with the system that presented the antigen to the T cell. There's communication between the infected cells in Th2 to the antigen-presenting cells that take it to the CD8 T cells, and the CD8 T cells will express the appropriate chemokine or CXCR3 that will identify the cell that is infected, and that's what allows them to migrate. They bind now these T cells, and they actually are pulled into the site of the infection, and that's how they produce the killing of the danger signal. This killing by the activated T cells now that have been differentiated and expanded, and they are in Th2, they could be CD4 dependent or CD4 independent. The CD4 T cell dependent pathway will present the antigen and activate the antigen for the CD8 primary response to deal with them. But there are other antigens or other danger signals that are independent of the CD4 signaling. And in this case, they will actually just deal with them directly with the CD8 T cells. In order to explain this a little bit more, I want to refer to the MHC class 1 and MHC class 2. So this is the way that the antigens will be processed. Depending on the antigen presenting cell will actually be the one that is infected by this virus or this danger signal. So this danger signal goes into a cell, and if this particular antigen is presented by the MHC class 1, the MHC class 1 interacts directly with the CD8 cytotoxic T cell, and it will produce a killing of the infected cell. And again, the presentation via the MHC class 1 depends on the antigen and on the cell that is actually digesting that antigen. And it directly causes the killing of that infected cell. On the other hand, other type of antigens are processed and presented by the MHC class 2. MHC class 2 is a bit more complex in the way that it actually will engage now the CD4 T cells. And these CD4 T cells that are engaged by the MHC class 2 with an antigen, what they do is they secrete cytokines. These cytokines will activate the macrophages that have the virus or the pathogen inside, and they will activate it to destroy it. So it's a different mechanisms. CD4 T cells are not killer cells, but they stimulate the other cells to kill it. CD4 T cells can also be engaged via the MHC2 by B cells. So when this particular antigen is seen by the B cell and processed by the B cell and presented via the MHC class 2, they will now engage the T cell. And likewise, the same as before, these CD4 T cells that have been presented an antigen via the MHC class 2 will produce cytokines that stimulate, in this case, the B cells into proliferation, maturation, and production of plasma cells that will now generate and secrete antibodies that are specific to that antigen. In the MHC class 2 response, you can have a combination of the two. You can have the direct activation of macrophages together with a B cell response. It's what we can have a cellular response and a humoral response by presence of antibodies. But again, depending on the type of danger signal and depending on the type of activation and presentation to the T cell, we have CD4 dependent responses or CD4 independent responses, where the major killing cell is the activated CD8 T cell that was migrated and that was actually engaged original in the lymphoid organ. Now, a very important thing of the immune system is an internal clock. This internal clock is a very interesting pathway where, interestingly, is precisely these CD8 active T cells the major producers of a single cytokine, which is called interleukin 10. Interleukin 10 is basically produced in the peripheral system, in the peripheral tissues, by the CD8 T cells, the same ones that produced inflammation. But it is very important to say that publications have more and more identifiers for where are the genes for IL-10 and which cells can actually synthesize IL-10. And I don't need to read you the complete list because there's growing and growing, but basically they can be Th1, Th2, Th17s, Treg cells, they could be B cells, NK cells, macrophages, dendritic cells. What this is really telling us, the message behind this multitude of different kinds of cells having this single gene and the ability to produce this single cytokine, is how important this cytokine is for us to be able to live. While it's crucial to have an immune system that is intact and that can deal with danger, it is as important to have a mechanism that controls that immune system and brings it back to inactivity. So interleukin 10 is a critical cytokine that will control the response of the immune system. And there's still a lot more to know about how IL-10 works. But
the production of interleukin 10 by this cytotoxic T lymphocyte requires several different activations of this particular cell that really is telling this cell that there was an inflammatory process going on. So the first thing that I want to talk about is interleukin 2. Interleukin 2 is one of the major products of a Th1 response and activation. And the production of interleukin 2 by the CD4 active T cells in the periphery will be one of the triggers telling the cytotoxic T lymphocyte to start producing the interleukin 10, which will dampen inflammation. So it's a product of an inflammatory process that's actually needed to start producing this control mechanism. Another very important mechanism of activation that has to also happen for the cytotoxic T lymphocyte to start producing and synthesizing the interleukin 10 is that via the T cell receptor that is specifically identifying the antigen that's presented by the antigen presenting cell. So that engagement is also important. It's important that the T cell is actually very specifically geared to responding to a very specific antigen. And that's required also for this control mechanism, modulation mechanism we have to control the inflammatory responses. And finally, interleukin 27, which is a product of the cells from the innate immune system will actually be also a key factor in producing interleukin-10, which will help dampen inflammation and decrease tissue damage. This mechanism is triggered after a lot of the inflammation has happened, and this is the way the cell knows that this is an end stage. Now that I get all these products of inflammation and I can see them as a cytotoxic T lymphocyte, then I can start producing interleukin-10 to dampen that reaction.
First, the immune system is a system that helps us identify danger signals. It is triggered by the danger signals that are presented to the T cell, and the T cell responds by differentiating and developing different kinds of cells with different effects. These effector cells are better characterized as Th1, Th2, Th17, Th22. There are more Th helper cells, but I will focus on these 4 critical Th phenotypes in this talk. These phenotypes are going to deal with danger and are going to help the organism heal. However, in some circumstances, they can also be associated with diseases. So how is it that in the immune system, the naive T cell, which I have drawn here in orange color, how is it that it responds to danger? The danger signals are going to be presented by an antigen-presenting cell, which here I drew as a dendritic cell. Basically, this dendritic cell is presenting the antigen in light pink to the T cell. The T cell has a T cell receptor that will actually identify that particular antigen or epitope, and with the appropriate co-stimulatory signals will actually respond to that danger signal. For more details on these interactions, I have other videos on the YouTube channel. But for now, let's just briefly mention that depending on the antigen presentation, the antigen being presented, the T cell receptor that is engaging the antigen, and the specific co-stimulatory signal, the T cell and the environment will actually start producing interleukin-12 and interferon gamma that in turn will develop that naive T cell in orange, and now it will specialize it into a Th1 cell. The Th1 cell is a T cell that is characterized by the synthesis and production of a profile of cytokines that goes outside from the T cells and has impact on organ tissue cells that will react with a typical inflammatory response. Please note that in this Th1 first secretion, we have a product here which is interferon gamma. That was also one of the key drivers for that T cell into differentiating to Th1. In other words, there's a perpetuation and a positive feedback loop when you have the first stimulation by that antigen presentation into Th1s to increase that response. And for the inflammatory responses that we need to deal when we cut ourselves with burns, with different pathogens, there is an initial increase in that inflammatory response that is necessary to deal with danger. In some circumstances, this can also be associated with diseases, and these are diseases with inflammatory components like rheumatoid arthritis, psoriatic arthritis, ankylosing spondylitis, lupus, psoriasis, etc. When the antigen that's being presented and the T cell receptor and co-stimulatory signals produce interleukin-4 instead of interleukin-12 and interferon gamma, the stimulation for the naive T cell is into becoming a Th2 response, the Th2 cell will be characterized by a different profile of cytokine secretion. These cytokines, interleukin-4, interleukin-5, interleukin-13, interleukin-25, will then target the cells like basophils, eosinophils, mast cells, and B cells into maturing and target tissues, basically epithelial cells in the gut, in the respiratory tract, and on the skin. These are very inflammatory allergic reactions. Note that also one of the products of the Th2 is also stimulating further differentiation into Th2. These allergic reactions are essential for dealing with bee stings, mosquito stings, etc. But also in disease, they associate with diseases like asthma, atopic dermatitis, other allergic reactions that may include also reactions to drugs, and inflammatory bowel disease or IBD. Now, while the Th1 response can be considered as an inflammatory response, and the Th2 response an allergic response, they're not necessarily completely isolated and separate. There are some cytokines that can crosstalk between one pathway and the other. TNF-alpha is a pluripotent cytokine that is produced by many different cells, and it does produce a lot of inflammation, and it can actually also stimulate B cells. Interleukin-2, on the other hand, will inhibit the late stage of switching of the B cells, so it is not typical to see a B cell complete switch to the isotype IgE in inflammatory responses, whereas that is more typical of the allergic responses that we see in the Th2 response. Interleukin-6 also stimulates the B cells. In the inflammatory responses, you do see B cell stimulation driven by either TNF and or IL-6 in, for instance, rheumatoid arthritis, but basically the rheumatoid factor will be an IgM without further isotype switch that is typical of the Th2 response. Antigen presentation and the milieu and co-stimulatory signals now produce a more complex pattern of different cytokines, including TGF-beta, interleukin-1, interleukin-6, interleukin-21, and interleukin-23. The differentiation of that naive T cell is going to lead to the Th1 cell. This cell has been named by the predominant cytokine that it will be releasing, which is interleukin-17. Th17 cells also produce interleukin-21 and TNF-alpha. There's a relationship between the Th17s and the Th22 cells. These cells actually are different in human beings, although in mice, the same cell produces both IL-17 and IL-22. These two pathways in humans, even though they're separate, they're still related. And they have an effect on neutrophils and keratinocytes, especially on the skin. They also crosstalk with the Th2 by having effects not only of perpetuation of the Th17 response, but also crosstalking with epithelial cells, cells on the skin, in the GI tract, and also in the respiratory tract. Furthermore, Th1 responses, basically with the production of interleukin-2, will then inhibit the differentiation to Th17. Likewise, IL-17 will inhibit the differentiation to Th1. In addition, IL-17 has been shown to inhibit NK cell activation. So there seems to be some sort of control between these different phenotypes where one would be favored over the other, although there's still some crosstalk in the end organs that can produce diseases and different disease patterns that could have a mixed profile. The Th17 involvement in psoriasis has been demonstrated by the recent efficacy results seen when the IL-17 pathway is inhibited. However, IL-17 may also be involved in other diseases like rheumatoid arthritis, psoriatic arthritis, inflammatory bowel disease. The picture is complex because these diseases were typically considered driven by Th1 responses, but we now can see that they could also be driven by Th17, and the explanation of how the two pathways can occur at the same time when in vitro one seems to inhibit the other are yet to be further characterized.
A final step in the development and differentiation of the naive T cell is when the environment driven by the interaction of the dendritic cell and the co-stimulatory signals plus the rest of the milieu that has already been producing and differentiation with different cells will actually be characterized by the production of TNF alpha, interleukin 6, interleukin 2, interleukin 27. And as you can see, this can come from the other pathways that have already been stimulated in the other differentiation. So this particular step in the milieu is a later stage where the predominance of these cytokines, either driven from a Th1 or a Th17 environment, or from the production of cytokines later on, like the interleukin 6 or interleukin 27, will stimulate a T regulatory cell that carries a FOXP3 gene. This particular cell is characterized by the synthesis and production of interleukin 10, also TGF-beta and interleukin 35. Interleukin 10 is the key, most instrumental cytokine for tolerance. In other words, these particular cytokines will actually be immunosuppressive, and it will dampen any other of the Th activated cell pathways that we have briefly reviewed in this video. So basically what happens is that independently of the particular response, was it a Th1 response that was seen either after we cut ourselves or any kind of inflammatory response to a particular disease, or was it a Th2 response after a mosquito bite, or a Th17 response, whichever that was, the milieu now is accumulating the cytokines TNF, IL-6, IL-2, IL-27 that will help in the differentiation of the T regulatory cells. I encourage you to see another section of the immune response videos that I have posted also on YouTube that have a little bit more of how this particular interaction occurs into the differentiation of this very important regulatory pathway. Why is this important? Basically, when we are dealing with any perception of danger, we have a first step, as I mentioned before, with positive loops that are retrofitting into an increase and enhancement. In other words, if we have a Th1 response, it will be producing interferon gamma that increases the Th1 response. If we have the Th2 response, we saw that IL-4 is both a product and also a stimulant for Th2. What happens is basically when we have any kind of inflammatory disease, we burn ourselves in the kitchen. Well, that particular blister and that particular response will eventually heal itself. Time heals all of these particular responses that we have, and it's mainly a sort of an internal regulatory system that we have with a T regulatory cell and IL-10. It is very interesting that some of the observations is why do we then have chronic diseases where there is a persistent inflammatory response that is not dampened? Well, there's several ideas and hypotheses that maybe there's no sufficient IL-10 or there's a lack of an IL-10 stimulatory reaction in these particular diseases. So either it be psoriasis or rheumatoid arthritis or inflammatory bowel disease or asthma, do we actually have a potential less efficient system for regulation? And of course, in this hypothesis, there's a lot of work being done in research on potential delivery systems, either delivering IL-10 to the systems and organs that have the chronic inflammation not controlled. Would that delivery of IL-10 help, or even stimulating this particular mechanism by using agonism systems for IL-27, IL-35, etc. So the role of these cytokines and all the interactions that they have is critical to understanding diseases and also understanding if we have a preferred choice of a particular treatment for different patients, which would be the cytokine pathway that we are inhibiting either with extracellular inhibition or intracellular mechanism for inhibiting these pathways, and what is the profile that we are seeing characterizing these diseases. I have several other videos in the YouTube channel where I have covered very briefly but comprehensively what would be the role, for instance, that we know in immuno-oncology of Th1s, Th2s, Th17s. How do we use different treatments in immuno-oncology to control this? And of course, I also have created some other educational videos that explain what is the cytokine signaling, how do we use it externally, what are the available armamentarium of drugs that inhibit particular cytokine pathways or particular phenotypes. Thank you for listening. As I mentioned, I have a series in my YouTube channel. I put it right there on the immune response. And I hope that you can take a look, check it, and see if this is useful. To create the different slides that you have seen and the different cartoons and animations, I have used many different references, but the key references are listed here, and they will be posted also in the YouTube description so that you can also take a look at them. And I hope you enjoy, and I hope this was useful. Thank you very much.